Hello, everyone, and welcome to today's live broadcast. Consideration is the use of analytical ultracentrifugation for characterization of AAV gene therapy delivery vectors. My name is Akash Bhattacharya, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Beckman Coulter Life Sciences. Please note that the Optima AUC is not intended or validated for use in the diagnosis of disease or other conditions. Also, the views expressed in this webinar are those of the speaker and not of Beckman Coulter Life Sciences. For more information on Beckman Coulter, please visit www.beckman.com/resources. Before we get started, we'd like to get to know our audience with a couple of questions. The first question, is analytical ultracentrifugation, or AUC, currently a part of your analytical workflows? Option A for yes, and option B for no. The second question, are good manufacturing practices, or GMP, important to your analytical workflow needs? Option A for very important. Option B for somewhat important. Option C for not very important. And option D for not sure. And finally, the third question. Your work environment is best described as option A for R&D or discovery, option B for process development, option C for manufacturing, and option D for quality control, analytical development, or characterization. Thank you all for your answers. With that, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. Also, please notice that you can share this webinar on your personal social media. Just click on the Social Sharing tab to let your friends and colleagues know about today's live event. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credit. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now introduce our presenter, Dr. Sucato, Associate Director of Biophysical Characterization at Charles River Laboratories. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Sucato, you may now begin your presentation.
Thank you, Akash. It's a pleasure to be able to present this webinar today on AUC and gene therapy, with a focus in particular in our Charles River Laboratories on AAV platforms. While in the past, several mechanisms for gene delivery have been advanced as potential therapies and have held the promise of a stable and long-term incorporation of vector DNA into host cells, it's safe to say that, at present, adeno-associated viral vectors, or AAVs that is, are indeed the leading choice for gene therapy, the treatment for a variety of conditions. AUC, meanwhile, and the size distribution profiles that derive from the sedimentation data are becoming more and more a key measurement in the AAV development process. What's contributed recently to the success of AAV-based strategies are some advances on the discovery and R&D side in developing recombinant AAVs with clinically advantageous properties, optimized transgenes that are efficiently delivered to the host cell nucleus and are reliably translated, and on the process and manufacturing side, likewise, advances there that have made AAV a practical drug material from a commercial perspective. These developments, scientifically, are of great value and interest, I'm sure, to this audience and to the field in general, while a deep dive into many of those aspects would be beyond the scope of this webinar. I'll present a collection of some references at the end for anyone who may be interested. Nevertheless, some of the recent advances in AAV technology can be understood in terms of the schematic, illustrating how good manufacturing can lead to a product with an improved profile in terms of the empty, full, and other species, a critical quality attribute, incidentally, which will be a focal point of this presentation, but also favorable capsid properties, including properties of the capsid shell, the VP1, 2, and 3 protein makeup, gained by recombinant expression optimization, which can guide the tissue specificity of initial receptor-mediated endocytosis, the endocytotic process overall, and the cytosol, and likewise favorable properties of the transgene, allowing for packaging of the maximal gene size and efficient, stable incorporation in the nucleus. The timeline shown here makes a good case for why discussions like these are particularly well-timed. It is only recently, after all, that clinical applications of AAV have begun to take off commercially. Development pipelines expanding in earnest really only in the last decade, and only 2 FDA-approved products at present, but with many, many more in development pipelines globally, and regulatory guidance on the so-called CQAs that define a safe and effective AAV product still taking form in many respects. In this talk, we'll discuss, and importantly show data also, in particular how recent advances in AUC characterization of AAV products may help inform that type of guidance. Now, in the past, at presentations like this, I've sometimes gone into some depth about the history of AAV, its wild-type traits, etc., that have led up to the present-day status of AAV as a gene delivery platform. In the interest of time, I'll shorten that part somewhat here, but suffice it to say, there are indeed plenty of review sources for this kind of historical background. Nevertheless, briefly, let me say that wild-type AAV, once thought to be simply a contaminant of adenovirus preparations, was first characterized extensively as early as the 1960s as a unique viral species. It was shown to be replication defective, going through its full replication cycle, only if the host was co-infected with a suitable helper virus, such as adeno or herpes virus. An important observation at the time was the finding that AAV itself only caused a mild immune response, such as the generation of neutralizing antibodies, and infection did not produce any observable disease condition. The apparent lack of pathogenicity in the wild type was a main advantage that would be leveraged in the development of recombinant AAV in gene therapy. Other advantages are related to the infection cycle of the wild type. For example, in the latent phase, AAV integrates itself stably into the human genome at only a single specific chromosomal site. And secondly, AAV has a good infectivity rate with quiescent or non-dividing cells. These advantages are in contrast to retroviruses, which may present a random insertion and mutagenicity risk, and are generally less effective in non-dividing cells. Now, the wild-type AAV virion itself is a small, non-enveloped icosahedral particle, about 20 nanometers in diameter. And one of the few disadvantages, incidentally, to AAV is this small capsid geometry and the relative size of the gene that can therefore be packaged. The AAV capsids contain a linear, in the wild-type AAV capsids, contain a linear single-stranded DNA genome of about 4.8 kilobase pairs. And wild-type AAVs are replication defective, as mentioned. In other words, they are dependent in the wild type on co-infection with a suitable helper virus, such as adenovirus, in order to complete their native replication cycle. Yet, recombinant AAV has several advantages as a gene delivery vector. It is safe. It's not known to be associated with any disease symptoms. It's effective, showing long-term transgene expression. And it's also quite robust in many cases, being known to be heat stable and showing stability under storage conditions, etc. Now, although the presence of a helper virus is a requirement for AAVs in the wild type, when developed for use as a gene therapy vector, production of recombinant AAV, not wild type, but recombinant AAV, can in fact be accomplished by helper-free methods, where the necessary helper factors are co-transfected into the production cell line. Additionally, in most production schemes, such as this quite popular and common triple co-transfection method illustrated here, the wild-type AAV genes for replication and for the structural capsid proteins are also co-transfected. The rep and cap genes are removed from the viral target DNA, and only the therapeutic transgene sits between the inverted terminal repeats, as illustrated in the capsid diagram. So there are a few different ways to accomplish this endpoint, and what I mean by that is the purified and concentrated end product, as illustrated at the bottom right of the screen. So it's this purified drug substance, as it were, that turns out, as it turns out, there's a few ways to accomplish this endpoint, the purified drug substance. And as it turns out, the exact modes of production and purification employed in the manufacture of recombinant AAV can have an important influence on attributes that appear to be critical to the quality of the final product. One such critical attribute is related to the nucleic acid that is packaged within the capsids of recombinant AAV. When produced from transfected cell lines, it has been shown that recombinantly produced AAVs are not uniform with respect to the packaging of DNA into the core. A high percentage of capsids may contain no genetic material at all and would be termed empty capsids, as shown on the left-hand side. Additionally, capsids have been shown to contain incomplete portions or fragments of transgene coding DNA, or even non-target extraneous nucleic acid contaminants. These species are illustrated on the right-hand side of the figure. So these observed empty or partially filled variants of recombinant AAVs can be dependent on the production and purification method and are a critical quality attribute because recent reports suggest that the extent of empty or partial capsids in an AAV drug product may correlate with vector-related side effects and reduced gene transduction rates, which are unwanted effects from a drug product. But yet, other reports hint at possible benefits derived from having at least some percentage of the capsids in the drug product as empties. So, as I mentioned in the very first slides, a big part of gene delivery characterization at Charles River is done by analytical ultracentrifugation. And I look forward in just a few moments to getting into some of the data and the discussion associated with that. But I feel I must spend a little time on the following question shown here, because it is something that comes up all the time in our business. The question of just how to treat these capsid-related impurities that invariably show up in AAV drug products, empty capsids being the most abundant in many cases. Essentially, though, how should the acceptability limits, the acceptable limit thresholds for these species be set? What's clear from guidance documents is that levels of empty capsids and other capsid-related variants should be closely monitored. That much is beyond question. Unfortunately, there is no one answer as to what levels of these impurities or contaminants would be considered an acceptability threshold across the board for all AAV drug products. Instead, product-specific assessment of the empty-full ratio for these specific products, as well as the presence of partial species, etc., should be done in correlation with toxicology and potency data obtained on the same sample type in order to justify any specific acceptability limits for a specific product line. While our focus in the biophysical group at Charles River is the validity of the AUC assay itself, we understand the importance of doing this work with some sense of where it all fits into the overall regulatory approval picture. And so I've included a few references here towards that end. This reference will certainly be of value to anyone interested in regulatory guidance. This FDA publication is from January of this year and is the latest guidance on CMC best practices, and in particular, section 32P223, as is excerpted here on the right-hand side, and the references and the discussion in particular within that section is important and gives the reader a sense of how empty levels should be defined and approached. Just how to think of empties, though, is still an active area of development, and this current opinion piece, still quite contemporary, presents some case for making the best of drug products that do contain significant levels of empties, how these products may still be moved forward, despite having empty levels that may be significant. Whilst here, in a review from Wright at the University of Pennsylvania, some of the downsides that empties may present to AAV therapies, and the case for keeping empties low for removing empties from drug products is covered. Lastly, here is a link to a very informative discussion that was part of a virtual think tank series recently. It has some very thoughtful insights into how regulatory thinking may evolve on the topic of gene therapy, hosted by some leaders with regulatory experience. And so certainly, regulatory thinking is evolving. AAV technology is evolving in terms of the production and purification strategies. So certainly you could say that evolution, or perhaps even revolution, is at hand, judging by the development pipeline for AAVs. While, as mentioned, only 2 AAV therapies are FDA approved at present, a great number of candidates will enter into clinical trials in the near future, representing a variety of AAV serotypes and targeting a wide range of disease conditions or target tissues, as these graphs make clear. Certainly, given the rationale that the species distribution profile of these diverse products should be carefully and individually assessed in accord with tox and potency data specific to these individual products, the importance of methods like AUC becomes apparent, which I think makes it a good time to show you just indeed what AUC can do. Now, the AUC assay itself is by no means trivial, and we could spend an entire webinar just on the fundamentals of the sedimentation process, instrument parameters, and the data deconvolution process. Suffice it to say, the new Optima model of AUC by Beckman-Coulter presents the opportunity for improved analysis, faster scanning, easy setup of dual absorbance and interference detection or multi-wavelength studies that can be done in a single experiment, and potentially improved signal-to-noise and resolution as well. Rotors and cell parts remain largely unchanged at present, and the fundamental data collected in an AUC experiment is shown here in this graph. The fundamental data is the sedimentation profile that's shown here, generated by spinning these special cells, the special AUC cells as illustrated, at high centrifugal velocity, causing sedimentation of heavy species like viral particles. Now, if the sample is highly monodisperse, a single boundary region or inflection will appear, as is illustrated in the trace just above the cell diagram. And the travel of this boundary will move from the top of the cell towards the bottom over time, that travel is a function of the rotor speed and the mass of the species that's being sedimented. Now, if the sample is heterogeneous, each of the sedimenting species in the sample may contribute to the raw data in the form of not just a single boundary or inflection point, but a superposition of several different boundaries, each proportional to the abundance of the individual species and the mass of that sedimenting species. Numerical fitting algorithms take this raw sedimentation data and deconvolve it to a peak distribution profile, such as the graph seen at the very far right-hand side, which we can readily observe and quantify. It's that final product, that final data that we can look at and quantify. Now, I've gone through these AUC basics rather quickly, I admit, in favor of focusing the remainder of the presentation on showing some practical and real results. Many resources, however, are available to dive deeper into the fundamentals of the assay and the fitting process, which I admit I have not talked about at great length, but a great review is one from Professor Jim Cole at the University of Connecticut, a paper from 2008, which is still very relevant and I must say a very good read, so I reference it here. But folks, here is what a real AUC experiment and setup. Here's what a real AUC experimental setup and result looks like. I should note that these data and all results I'll show hereafter derive from R&D model AUC samples. These are not data from clinical samples nor drug products, but are instead from simple R&D model samples that nevertheless are a good representation of AAV behavior. For this sample, which I will refer to as sample A, we loaded about 150 microliters at a titer of approximately 1 e to the 13th viral particles per mil. Centrifugation was at 20,000 RPMs, and detection was at 230 nanometers. This is an absorbance-only detection mode. As you can see, the sample concentration in the instrument was about 0.5 OD, and you can see that from the y-axis of the rainbow-colored curves in the panel labeled A. And we collected about 60 scans, which collected over the course of about 4 hours. These results are a series of sedimentation profiles that, again, collected over time, each trace representing essentially a single time point. And the data density, or that term data density, can be thought of as how closely spaced these individual traces are. As I mentioned, the movement of these boundary regions give you some sense of the sample heterogeneity and the relative size of the species. In the case of this sample, because there are apparently multiple boundaries present, clearly this sample is not monodisperse. There may be more than one species present in this sample. Contrast the results on sample A with those of sample B, which I show here. This sample is also an AAV. Incidentally, sample B is of a different serotype than sample A, but nevertheless very similar in many respects. This sample was run on the AUC in the same exact way, on the same AUC instrument, the same exact way as sample A, and we have a raw data set as shown here. Some subtle differences in the raw data do appear, as you can see if you contrast the data from A and B. There are multiple boundaries present in both, but the amplitude and the rate of travel might be somewhat different. Importantly, when the fitted results are observed, as shown here for sample A and here for sample B, the difference between the two is more readily interpreted, and the value of AUC becomes very clear. Observe here at about 80S, a peak which is an intermediate species, intermediate that is between the empty and the full capsid species which are labeled. This intermediate may correspond to a partially filled capsid species or some other type of variant. But nevertheless, what's clear from these two AUC assays is that the method is capable of resolving and quantifying the relative abundance not only of empty and full, which it does quite well, but even intermediates with excellent baseline resolution. And clearly, AUC can discern the difference between relatively small percent abundances, as I illustrate here in the partial peak, which has a very distinctive difference between its relative abundance in sample A and in sample B. Now these samples may be from two different serotypes, but if you are developing an AAV product and are comparing two or more samples as a result of a process change, etc., the difference between this result and this result may be significant to your development decisions. It may be very significant to your development decisions. And it's good to know that AUC can make these kind of distinctions, whether these be different serotypes or from different process changes, etc. While some other methods like capillary electrophoresis and ion exchange HPLC are indeed being developed for MT full resolution, at present there is no method that can surpass the kind of resolution demonstrated here for an AAV sample. Again, showing excellent baseline resolution, excellent and reliable baseline resolution, not only for empty and full capsids, but for intermediate species as well, and possibly aggregates too, towards the right-hand side, which we will get into. Nevertheless, the ability to resolve subtle differences in the abundance of small peaks, as I've just shown, would only be reliable if the method had sufficient precision. And of course, precision is a primary objective for any method validation plan. So we've investigated what is the expected precision obtained for AUC assays when applied to AAV. I show here a set of n equals 6 runs done by the same analyst on the same day, and the statistics for the sedimentation coefficient and the peak area indicate that precision by AUC is indeed quite fit for purpose. Both values, both S and percent area, having RSDs for those values that are quite respectable. Now, admittedly, chromatographic methods are able to obtain higher precision levels than shown here for AUC. HPLC may have improved RSDs over what's shown here, but the kind of repeatability shown here in both the statistical numbers in the table and the relatively good overlap between replicants, as evidenced visually by the graph showing 6 overlaid curves from the same 6 replicates from the same sample, this would tend to argue that AUC is indeed very suitable for quantifying empty and full levels, especially given the very good peak separation shown here, which is quite honestly difficult to obtain by chromatography. And the longer-term variability, as we've done, as shown here by intermediate precision, whereby the same sample was run, but on a second day, second analyst, a second setup, again supports the notion that that the AUC, when applied to AAV samples, will have the kind of precision both in repeatability and longer-term intermediate precision that you can count on for a validated method. Another parameter that we've evaluated in our quest to determine if AUC can meet validation standards is linearity, which I'll cover here. We've actually evaluated the linear response of AUC in two different ways. First is by the means I show in this slide, where we have run AUC on a series of samples. We've created the series by starting with the so-called full capsid stock, as shown in the lighter purple color on the left-hand side. We've used that as a baseline, and then titrated in known amounts of another stock, which is enriched in empty capsid. That's the darker purple color on the right-hand side of the schematic. By adding known amounts of empty to a fixed and also known amount of full capsids, we have the ability to challenge the AUC assay in its capability to have a linear response to a change in the amount of one species relative to another, which remains relatively fixed. And the data shown in the graph indicates that while the species at 80 and 100S remain relatively fixed in their peak shape and size, their percent abundance, the area under the curve for the empty capsid increases linearly in correspondence with the amount of empty capsid spiked in. The data indicate here that indeed a good linear response is achieved. And this is a significant accomplishment for the AUC assay because this result, together with a reasonable value for precision, gives the analyst confidence that small differences in the empty-full ratio will be picked up by the instrument. A variation of the linearity challenge to AUC is the ability to show a correlation between the total integrated area as a function of the overall amount of sample loaded into the cell. And this is what I illustrate here in this slide, where we start with a stock of that full sample and then just do serial two-fold dilutions downward from there. We've evaluated that kind of situation by taking, again, as I say, that stock amount of sample and making the serial two-fold dilution series. When we run that sample set in the AUC, again, we see a nice linear response, a nice linear trend between the overall concentration loaded in the cell and the total integrated area under the resulting distribution, as the graph shows, in area units. As I say, the curve for all integrated species moving downward linearly, the total integrated area having a nice one-to-one correspondence between that measured area and the amount that's loaded. This is good because it shows the concept of recovery, which holds true for AUC in much the similar way that it would for chromatographic assays, where one often does a sort of a mass balance standard curve in chromatography. So this is a reassuring result in AUC. Knowing the limits of detection in an AUC assay will also be important to method validation. In HPLC, the LOD and the LOQ would often be defined by the signal to noise, such as the signal to noise in the baseline of a buffer blank, where the peak of interest would normally appear. In AUC, the baseline of a blank scan is artificially smooth and very low in noise because the CFS distribution, the kind of graph that we're looking at here, is really a fitted result. This is not the noise from a detector. This is the algorithm's attempts to fit species in a certain region. And if no species are there to be observed, the noise is somewhat artificially low. Therefore, defining LOD and LOQ by this method would tend to lead to values that are perhaps not really realistic for a limit of detection. It might be artificially low. Because the sedimentation profiles, or the raw data, should ultimately define the resolution of the presence of multiple species. In other words, from the ability to resolve different boundary regions or inflection regions in a curve like I show here. The noise in the raw data should be a strong influence on the ultimate limit of detection of species in the fitted distribution profile. In AUC, in Charles River, we have calculated LOD and LOQ by testing a series of standards at low dilution, so very low dilutions of the stock sample, in a manner similar to the slope response analysis that one would do in HPLC. Using that method, we find limits of quantitation on the order of about 1%, which is consistent with the noise level in the raw scans like those shown here. Part of any good validation will also be robustness, the demonstration that the method can remain valid even despite some usually small perturbations in the method runtime parameters. Or if robustness is to fail, then within what limits can robustness still be found? We challenged the AUC assay in this regard with respect to these method perturbations shown here, and we found the following. With respect to the alignment of sample cells in the rotor, there really is very little room for error. But we at Charles River have an alignment tool for the purpose of cell placement in the rotor, and when used as directed, data meets precision expectations in accord with what was shown several slides earlier in the presentation on precision. However, if some amount of misalignment is intentionally introduced as part of the robustness perturbation, we find that even very small misalignments, as small as 1 to 2 degrees, will lead to unacceptable data. Sensitivity with respect to this parameter is perhaps not surprising, since it has been reported for protein-based systems in earlier work, and I show some references here. But we find in this work, for larger species like AAVs, which sediment relatively fast, the measurement is especially sensitive to misalignment, and there really is very little tolerance for misalignment in AUC work on AAV samples, an important finding that we've discovered in our studies. With respect to the time allowed for temperature equilibration, that is the time the rotor is allowed to sit still or idle in order that the rotor body and the samples reach a temperature uniformity throughout prior to spinning, we do find the method to be robust for wait times greater than about 60 minutes. Equilibration times shorter than this can lead to unstable data. Therefore, wait times greater than about 1.5 hours would usually be recommended for any regulated work. The preset centrifugal velocity parameter, shown next, is generally quite robust. When we dialed in spin speeds that differed by plus or minus 10% of the nominal target velocity, results at these slightly different spin speeds still matched the expected results from the nominal situation. Lastly, for robustness, we have the scan frequency parameter, which I must say is still a very active area of research in our laboratory. While we have yet to fully evaluate the effect of different scanning frequencies, or in other words, the different data density profiles, the density of these individual profiles that make up the raw data set, as you can see from the rainbow-colored curves, how densely those are packed. We do know, however, that occasional scans that may be missed, such as an occasional skipped scan that may occur by the instrument, these occasional skips do not perturb the data set to any significant degree. That is to say, the assay is still robust for this type of occurrence and the occasional skipped scan by the instrument. Another important finding to come out of this work. And so the presentation thus far has been geared towards showing how the AUC assay can meet many, if not all, of the criteria of a validated assay, having reasonable results, as I've shown, for precision, linearity, and so forth. Results that make the case that the AUC assay is indeed fit for the purpose of judging the empty-full ratio and other properties. But one overarching characteristic of the AUC assay that must be present these days is the data integrity associated with the measurement of the electronic records and the downstream analysis. Increasingly, the expectation is that the principles of so-called ALCOA, attributability, legibility, etc., illustrated here, that these principles are being held to for any assay, but certainly for those assays that are to be used for lot release or on clinical samples. Can the electronic records of an AUC assay attain these goals? The AUC assay has not had the head start in this area that other assays like HPLC have had. AUC, of course, has primarily been a strictly R&D tool for most of its history, but it's been a very important tool. It's been a critical part of life science research for many decades, but it has never, until now, really been needed in a regulatory compliant way. With enough depth, however, any gaps we feel can be overcome, and we at Charles River have overcome these gaps, if one has computer systems and IT resources to leverage and create workarounds where gaps do exist. And I'll list a few such solutions that can be put in place to lend some credibility, some reliability and compliance, I should say, as I show here in this table, to the data handling side of the AUC endeavor. Importantly, as I show here in a few bullet points, CSV and IT groups can implement layers of security through add-on scripts to ensure accessibility restrictions to the software, adding a layer of security where the software may presently be lacking. The custom scripts can also be added to make sure primary raw data, once generated, is automatically moved and is stored read-only in a way that requires very little intervention or interaction from the analyst, the one who is logged in on the computer. We make this easier and secure through custom scripts. But where software gaps cannot be bridged by electronic solutions, we have reverted to paper-based methods where we need to, including review checkpoints built into the protocol and a robust data form-driven process for reporting, which can show compliance literally every step along the way. Now, while the paper trail that may result from work like this can be somewhat cumbersome, especially in an age where nearly everything is electronic, the compliance nevertheless is there, which we've proven. All this can be done as we have, although not without a good deal of effort on the part of the executing laboratory. Reassuring, perhaps, though, is the notion that in the near future, soon, many of these aspects of data integrity may come built in to the software, making GMP-compliant AUC more of an off-the-shelf reality. So that being said, I'll round out the data portion of this presentation with a few other ways, beyond what I've shown, in which AUC can also be applied to AAV samples. The data portion of the talk so far has been focused on the advantages of AUC as a means to characterize the empty full distribution, or the abundance of other species such as partials that may be present. But beyond that, the benefits of AUC can be extended to other AAV applications as well. Specifically, dual wavelength experiments can be done, as I'll talk about, and there's some richness of information that can be gained there, as well as aggregation. Aggregation can be measured apart from the conventional distributions which I've shown, which I'll also cover. And so conventional empty full characterization is usually done at a low UV wavelength like 230 nanometers, where absorptivity is very strong, and where there is likely to be relative parity in terms of the absorbance strength of empty and full capsids. However, as is well known, at longer wavelengths, we have some difference between the absorbance properties of proteins versus nucleic acids. That is to say, aromatic protein side chains clearly having an absorbance peak around 280 nanometers, while nucleic acids showing a much stronger signal lower at 260 nanometers. So we can take advantage of this disparity between proteins and nucleic acid, and also take advantage of newer AUC instrumentation, which makes dual 260 to 80 detection easy to do in a single experiment. So by doing so, we can determine in a single simple spin if a certain peak is likely to be associated with nucleic acids or not. In this example, which highlights the differential A260 and A280 signal of the empty peak, as I show here in these two graphs, which I toggle between, you see a much weaker A260 absorbance for the empty peak compared to the A280 absorbance from the peak that derives from the same species. Now, this is expected for empty capsids, since they should in principle be all protein and contain no nucleic acid. So the A260 signal is really just residual, very small. But the same principle can be applied to other regions of the distribution, where it may be important to have some idea of what the capsid identity, what the species identity is. Now, the data I show here is on a well-behaved model system, but some products that we deal with have a good deal of heterogeneity around the main peak at about 100S, as I show in this graph. Some samples may show a lot of broadness in this region, and in some cases have resolvable shoulders off of what is ostensibly the main peak. So the identity of these kind of what this broadness or what these shoulders may correspond to is a matter of some question. In these cases, it may be very worthwhile to run a dual 260-280 assay to investigate how the signal varies between those two detection wavelengths, which may shed some light on the source of heterogeneity in this crucial region. And so beyond just heterogeneity in the neighborhood of the empty and full peaks, another important characteristic of an AAV sample will be the extent of aggregates that may be present. While other methods besides AUC have traditionally been used to measure aggregation, notably size exclusion HPLC and dynamic light scattering, When it comes to aggregation in AAV samples, AUC may have some advantages over these other methods. Specifically, an AUC separation has no stationary phase or column matrix as does SEC. High molecular weight species, which may be present from capsid aggregation, can be trapped by HPLC columns and therefore missed by these types of assays. Now DLS, like AUC, has no stationary phase, but DLS, dynamic light scattering, makes no separation of the capsid species, and partly for this reason, any size distribution information which may be obtainable by fitting DLS data is lacking in the kind of resolution that an AUC assay can provide. The data I show here was obtained by subjecting an AAV sample to fairly strong thermal stress, as indicated in the graph, indicating that if aggregation does occur, it is over a fairly large size range, many hundreds of S values as indicated in the figure. As demonstrated, AUC has the ability to quantify aggregation over this very large size range. Not only that, at the same time, it's able to resolve some very real peak structure over this range. And that's a capability that may not be possible with other methods. That brings me to the end of, we're nearing the end of the presentation. And so, in conclusion, what I've demonstrated in today's webinar is that the AUC assay, when applied to the characterization of recombinant AAV products, one, provides excellent resolution of empty, full, and other AAV product variants on par or even superior to other methods. It's capable of meeting many of the criteria for, many if not all, of the criteria for a validated method, including precision, linearity, and robustness parameters that make AUC fit for the purpose of quantifying heterogeneity in AAV clinical samples. 3, with the proper QA and CSV resources, the instrument and the assay can be made to comply with data integrity standards. And lastly, with the right labor force and instrumentation, and with the right amount of personnel and investment in hardware and instrumentation, AUC can be run at a throughput suitable for most development needs. But not only that, AUC can do much more. It's very versatile. You can do now with the new Optima multi-wavelength studies very easily, and studies on aggregation, making it a very competitive technique overall when it comes to gene delivery products. In closing, I'll add just a few little bits of information in a few graphs illustrating the quite high titer levels presently achieved in the production of more conventional protein-based therapeutics. Some of these products, in fact, showing at least a modest improvement in titer over years of production. Again, these are for traditional protein-based drugs, which have been around much longer than AAVs or other gene delivery vectors. Remarkably, given the demand for some of these products, which are now approved and produced at commercial scale, the raw amounts of these drugs in terms of production can measure in the hundreds of pounds or more annually, which is quite a remarkable statistic. Will recombinant AAV products someday also be produced in these large amounts? If so, then certainly characterization methods will have to be up to that challenge. With this kind of market potential, and with today's instrumentation allowing for improved data quality and throughput, if you are in the field of gene therapy, consider asking what AUC can do for you and for your characterization needs. At Charles River, we are here to help in that respect, and also for any other questions pertaining to biologic testing. My contacts are shown here, and I'm happy to be of service. I would ask that you also include this address as well for proper routing. I'd like to give a special thanks to the following individuals: Kalala Biswas, the Site Director for Biologics in Shrewsbury, Massachusetts, as well as the AUC team consisting of Michael Rivet, Nicholas MacArthur, and Vi Vu, who've done many of the experiments that form the basis for this work. The other members of the biophysical group you see listed here. And finally, thank you for listening. It's been a pleasure to have been your host for this presentation. just a few final slides. Here I have the references that I've mentioned throughout the talk. And lastly, some disclaimer information. That concludes today's presentation. Thank you again, and good day.
Thank you, Dr. Sucato. So this is for the audience. While we take a moment and allow Dr. Sucato to catch his breath, we would like to put a question in front of you. So the question is as follows. We would like to know if AUC is a future consideration for your laboratory needs. Please select one of the following options. Option A, not applicable, I am already using AUC. Option B, yes, and I would like to speak to a Beckman Coulter sales specialist. Option C, yes, but I prefer someone else runs my sample. Option D, maybe, but I'm still unsure whether AUC is a fit for my sample. And option E for no, I do not foresee a future need for AUC in my laboratory. Thank you, Dr. Sucato, for this extremely informative and very in-depth presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We will answer as many questions as we have time for today. So let's get started. The first question we have in is as follows. This is a question about experimental consideration. How much AAV sample would you need to do a basic characterization assay? And how much would you need for a full validation? Over to you, Dr. Sucato.
Thank you, Akash. Yeah, that's a very good question. And it's a question we get many times from people that need to get into AAV studies for the first time. We are used to working with AAV titers that are in the range of about 1 e to the 13th viral particles per mil. It's a common titer level that we work with. If you can have titer levels that high, we only really need about 100 or 200 microliters to do a routine R&D level test, and that would get you empty full characterization, the presence of any partials or any aggregates in the neighborhood of the empty and full species. Again, that's about 100 to 200 microliters, and that would get you duplicate analysis in a single centrifugal experiment. Now, a method validation, as I've shown, involves many parts, many parameters are evaluated, and so there's many assays that comprise a full validation. For that reason, the amounts required there are much more, on the order of 5 milliliters at that same titer level of about 1 e to the 13th. Now, while that might seem like a lot, we've noticed over the course of several years that AUC has become an emerging method for AAV drug developers, that the amounts that are being able to be produced from improved manufacturing and process improvements make that more and more of a reality, those kind of amounts. So we hope to see more process improvements in the future that will allow these kind of methods to go forward.
Thank you, Dr. Sucato. So the second question is very much in the same context. Does Charles River Laboratory also perform any orthogonal or complementary assays for gene therapy products such as AAV?
Yeah, we do. At Charles River, we have a few complementary methods that are available and are known to the field in general. Some of these are older techniques that still have some utility. Transmission electron microscopy, or TEM, has been traditionally used to characterize AAV products and produces some nice image-based results. That's still done to some extent, but as the field advances, AUC is quickly becoming the method of choice, as I say. is really becoming the front runner or the gold standard for empty full characterization. Yet, other assays are receiving a lot of attention and a lot of R&D and optimization efforts, including capillary electrophoresis-based methods, including CIEF and isoelectric focusing, as well as ion exchange chromatography. Now, these methods may have some improvements in the very near future that put those methods in terms of the baseline resolution that they can achieve potentially on par with AUC. At present, we know of no method that can surpass AUC in terms of the data quality, the separation of these peaks, the empty and the full, the baseline resolution, and the good signal to noise.
Thank you, Dr. Sucato. So we have two related questions coming up, and they are both on the analysis side of things. So the first of these questions is as follows. If a new commercial software was to be developed for the analysis of AUC data, especially focused upon gene therapy samples, then what kind of computing modes or models would you like to see implemented in such a software?
Thanks, Akash. Yeah. So in terms of the software, and if these software offerings become widely available in a industry-friendly, GMP-friendly way, then certainly the types of fitting models I would like to see are something akin to the traditional CFS distribution, which is a standard component of SEDFIT, which is a very popular algorithm. So clearly, a CFS model or a CFS-like model would be an important factor in anything that we would look for. Not only that, but some models that are capable of explicitly entering in discrete species. A CFS model makes no prior assumptions as to how many peaks may be present, but some more advanced models might give you the option of adding in prior information about where certain peaks might show up, which can aid in the fitting process. So that would be a nice function to have, as well as fitting models that can fit simultaneously both the sedimentation coefficient as well as information on shapes. So sedimentation coefficient is very much a mass-based, very much correlated with the mass of the species, but there's also some shape information which may be embedded within the AUC data. So a fitting algorithm that can make simultaneous sedimentation coefficient and frictional ratio or diffusion coefficient information available from a single fit would be an excellent attribute, I think.
Thank you, Dr. Sucato. And the next analysis question, which is very much on the lines of GMP, which you have mentioned a couple of times, is this. If such a commercial software was to be developed, then what kind of GMP features would you like it to have?
Yeah, that's great. And clearly, any kind of software offering in that regard, it should model what's present in chromatography, for example, having password-based restrictions to entering into the software, having audit trails for any changes that might be made to a method or fitting parameters, etc. Having that auditable where the user is required to acknowledge any changes, and that's tracked. The ability to lock methods, as well as data encryption, so encryption of the raw data, and proper storage or maybe even server-based filing of this information would be very worthwhile, I think.
Thank you, Dr. Sucato. And thank you to the audience for asking these questions. Those are all of the questions that we have time for today. I want to remind our audience that those questions that we were unable to answer today and those that come in during the on-demand availability period for this webinar will be answered via the email address that you provided at the time of registration. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Again, the questions that we were not able to get to today will be addressed by the speaker via the contact information that you have provided at the time of registration. We would like to once again thank Dr. Sucato for his time today and his valuable contributions to the AUC field. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you and goodbye.